autogamy. The other name for this self-pollination is selfing, okay, or it is inbreeding. Now we know that during the process of self-pollination, you can say the characters that are coming from the same type of plant, right? So the offspring will be the exact copies of the parents. There will be no new characters introduced into them. Okay, no new characters will be introduced. So when continued self-pollination is done for several generations, it leads to a phenomena that is known as inbreeding depression. Okay, so continued self-pollination. Always remember, only the continuation of self-pollination. Okay, it would the continued self-pollination leads to inbreeding depression. Okay, when like for several generations, not just one. Like it doesn't mean that the plant did the self-pollination once and it will undergo inbreeding depression. No, when continued self-pollination is done for several generations, then that will be leading to inbreeding depression. Now, how do we protect the plant from the inbreeding depression? We protect it from the inbreeding depression by outbreeding devices. Okay. Now, what are outbreeding devices? Self-pollination is inbreeding. Continued self-pollination leads to inbreeding depression. Now, outbreeding devices are the devices or features of the plants which discourages the self-pollination and encourages the cross-pollination. Now, does anybody here know why cross-pollination is advantageous than self-pollination? Any idea? Why cross-pollination is advantageous or more advantageous as compared to self-pollination?
Because in cross-pollination there is variation.
How? Yeah, you're right. Absolutely correct. How?
Yes, there are two different.
Parents.
Exactly. Because the two different, like the source of pollen is different, right? Because here of the different plant and the different flower. So the pollen which is coming can be from a different flower or a different plant all in together. So it brings along its own characters to the female part, right? That is why always remember that the cross pollination is 100% much more advantageous as compared to the self pollination. Also because of the reason that because of cross pollination, variations are introduced, and since variations are introduced, it would lead to evolution. Okay. So outbreeding devices are, see, devices does not mean anything. Devices means the mechanism of a flower. How does it perform in order to lessen the amount of self-pollination taking and increasing the amount of cross-pollination taking? So let us see what are those things. The first thing is unisexuality. We know that for bisexual flowers, wherein one flower contains the male and the female part in one only, automatically it will undergo self-pollination because the male is there, the female is there. The production of unisexual flower, either the flower will contain one male part or it will contain the female part. So there will be no other part, right? So unisexuality actually stops or discourages the self-pollination process. Now, talking about the two cases that comes under unisexuality, first is monoecious and then there is dioecious. Talking about monoecious, male and female flowers are produced on the same plant. The same plant has the male and female flowers, but the flowers are different. Talking about dioecious, male and female flowers are produced on two separate plants. This will prevent autogamy and geitonogamy. See, monoecious plant, if one plant has the male flower and it has the female flower, so we can say that 100% it can prevent the self-pollination, that is autogamy. But will it prevent geitonogamy? No, right? Because what happens during geitonogamy? The transfer of pollen takes place from the anther of a flower to the stigma of another flower, but the plant is same, right? So for monoecious, okay, we understand that, okay, self-pollination is being prevented, autogamy is being prevented, but geitonogamy is not prevented. So even though this prevents autogamy at some level, so we can count this as a device for outbreeding. And the example for the plants that actually have the monoecious flowers, it is castor and maize. Now talking about the dioecious, wherein the male and the female are produced on the two separate plants. One plant which has the male part, one plant which has the female part. Now this will prevent the autogamy as well as the geitonogamy. And what is the only way of pollination that can take place? That is xenogamy. Xenogamy means cross-pollination, right? And this takes place in the case of Vallisneria and papaya. So the first outbreeding device is clear to you people? Now, if I say monoecious or if I say dioecious, I mean unisexuality only, right? Because the male and the female flowers are different, whether on a different plant, whether on the same plant. So unisexuality is clear to everyone?
Yes.
Okay, very good. Moving forward, there is another type of outbreeding device, and this is known as dichogamy. It refers to the different maturation time of androecium and gynoecium in the same flower. See, first of all, there are so many gamy words in this chapter. So don't get confused. You know? Pay close attention to those things wherein what gamy is referring to what. Now talking about dichogamy, see, in order for a successful pollination or for successful reproduction to happen in the case of plants, the male and the female reproductive whorls should be matured at the same time. If the male is not matured but the female is matured, then who will give the pollen? But if the female is not matured and the male is matured, the pollen will come, but the fertilization will not take place. So the male and the female, their maturation has to be synchronized. But the opposite of that will prevent the self-pollination. If the male and female, if present on the same flower, if they are not maturing at the same time, it means that they cannot reproduce, isn't it? So the case one, which comes under dichogamy. Now, either the pollen is released before the stigma becomes receptive. Receptive means ready to receive the pollen. This case is known as proto-androus, wherein the pollen is released, meaning the anther got mature and the pollen was released. This is known as proto-androus. Proto means first. And androus is referring to the androecium, right? The male part. And this happens in the case of sunflower and cotton. Clear? This case one is clear to you people? Okay.
Yes.
Now, okay, very good. Now talking about case two. Now, in this case, the female part would become receptive, meaning ready to receive the pollen, but the pollen was not released. This is known as protogynous. Proto again means first. Gynous refers to the gynoecium, the female part. This takes place in the case of ficus and aristolochia. Okay, from now on, there will be many examples. So, dichogamy is clear to you people? Both the cases are clear? All right, very good. Now talking about self-incompatibility. Self-incompatibility, like the name suggests, even if they mature at the same time, even if they are perfectly fit for self-pollination or autogamy, but still, if in one flower, the male part releases the pollen and it lands on the stigma, because of self-incompatibility, this pollen will be rejected. Okay, so again, it will not lead to fertilization. So it is the failure of self-pollens from fertilizing the ovules by the inhibition of pollen germination or the pollen tube growth. Meaning, if this is the stigma, okay, and the pollen was released of the same flower, okay, self-pollen, that is why we're calling it. Now it lands here. Now, after the pollen grain lands on the stigma, the next step, normally what happens is that pollen tube growth takes place. Okay, for example, this is a pollen. So from here, a tube-like structure will grow. Okay, this phenomena is known as pollen tube germination or pollen germination or pollen tube growth. So if the pollen is a self-pollen, meaning of the same flower, then this will be inhibited. It will not be favored. And this happens because of, this is a genetic mechanism. Genetic mechanism means you can say the chemicals will not allow it to germinate. Clear? Any doubt? Why do you people look so confused?
But self-pollination happens in the same flower.
Yeah, self-pollination happens in the same flower. But see, there are certain species wherein they don't want their species to decline. So nature has it in them that their own pollen cannot fertilize them. Okay. They are ready for any like foreign pollen to come and fertilize, but the own pollen has given no value. Their pollen will fertilize some other flower, not theirs. So that is self-incompatibility. Incompatibility, you understand? Yes. Now, now in some species, the anther and the stigma are placed at different positions so that the pollen cannot come in contact with the stigma. Now, this is a very unique mechanism as well. They are compatible. It is a bisexual flower, but they are placed in such a way that even after the dehiscence of anther, the pollen will not fall on the stigma of the same flower. It will go elsewhere. This is the fourth mechanism. So all these four mechanisms or the outbreeding devices are clear to you people? Any doubts? Anything that you did not understand?
Can you explain the first one?
Which one?
The
Unisexuality?
Yeah.
Okay, yes, I can explain. Why are you scared? You'll ask me to explain it 10 times, I'll explain it 10 times. Okay. Unisexuality. See, when we were talking about pollination and we studied about autogamy, right? So we said that it is the transfer of pollen grains from the anther to the stigma of the same flower, right? And the second case was geitonogamy, in which we said that it is the transfer of pollen from the anther to the stigma of a different flower but the same plant, right? These two things you keep in mind. Now, if we are talking about the first case, that is autogamy, so we are referring to a bisexual flower if the flower is same and in one flower the male and the female part both of them are there, so it will cause self-pollination, right? So the best mechanism, the basic level to prevent that to happen is to produce the plant will produce flowers that will not be bisexual, right? It will produce unisexual flowers. Uni means one. Sexuality is referring to either the male or the female. Now, unisexuality has two cases. Monoecious and dioecious are the cases of unisexuality in plants. This is not the case in animals. In plants, monoecious and dioecious comes under unisexuality. Now see, monoecious means the male flowers and the female flowers are produced on the same plant. If this is my very beautiful pot and this is my plant. Okay, so here is the male flower. Here is the female flower. Now tell me, is this bisexual condition? Yes or no? Is this condition that I'm showing, is it bisexual condition? You people are getting what I'm trying to say. Bisexual is this.
In the same flower, there is male and female.
No, this is not bisexual.
This is bisexual. Do you remember that hibiscus flower that I had a picture in?
Yeah.
That is a bisexual condition. Not this badly drawn, but this is the condition, right? One flower will have the male and the female part. But here I'm saying this is one plant. One plant has separate flowers for female, separate flowers for male. This condition is monoecious. Okay, monoecious means plant is same, flowers are different. Okay, Now talking about the dioecious condition. This is one beautiful plant. This is another beautiful plant. This has only male flowers. This has only female flowers. Now this is dioecious. Two different plants. One has only male flowers, one has only female flowers. Now, in this case, if this is the male and that is the female, so the pollen will be transferred from the male to the female. It can be, right? So, is it preventing autogamy? Yes or no?
Yes.
100%, right? It is
Yeah.
preventing autogamy because the pollens are being transferred from another flower, like from one flower to another flower. But is it, what do you call it? What did I say? Is it
Preventing-
inhibiting geitonogamy?
No.
No, right? Because the source is same, right? It is one plant, right? So this monoecious case is good. It is, yes, it will be considered as an outbreeding device, but not so efficient. Then coming here, now they have no other choice. The male can't transfer to male, right? It has to transfer to female. This is one plant, this is another plant. So this is dioecious condition. It will prevent autogamy, it will prevent geitonogamy as well. And what will it actually?
Xenogamy.
Xenogamy. Xenogamy's other name is?
Cross-pollination.
So this is clear?
Yes.
And then these are the examples. Don't confuse between the examples. Any other thing? This
No.
dichogamy is clear to everyone?
Yeah.
What about
Yes.
self-compatibility?
Yeah.
All right. Now we have discussed every single thing up till, like starting from the formation of pollen up till the transfer of pollen. Meaning now the whole entire pre-fertilization event has been over. Now we are moving towards the fertilization part. Because ultimately when the pollens
How
have
many
been,
more topics are left in this chapter?
it is a long chapter. Very long. The first
In my school, they finished the second chapter too.
the second chapter as well.
Yeah.
I am teaching you in depth. Did they teach like this?
Nah.
Okay, see, this chapter is long, but why do you find it boring? I don't find it boring. You find it boring. It's good.
Kind of.
It's easy chapter. You'll get
Yeah, now it's fine.
good marks. Like it's very boring. It's not like, don't call it boring. I gave you full rights to call anatomy boring. I'll not give you rights to call this boring. And you know, like, what is the chapter after this? Is it human reproduction?
Yeah.
Yes.
Even that is long. So be ready for that also. Even that is long. Okay. All the chapters of, I think 12th are long only. Some chapters are short, but most of the chapters are long. But they are very scoring. Like you cannot miss 1 marks in this chapter. Okay. Now see, as I told you that when the pollen would land on the stigma, a receptive stigma, always remember, a receptive stigma, meaning a stigma which is like, come on, pollen, I'm ready. Okay. So a receptive stigma, whenever a pollen will land on it, it will release into, like, see, there, as the pollen is landing on the stigma, there will be a chemical dialogue between them. Okay, a chemical dialogue. It is known as pollen pistil interaction. Okay, a pollen pistil interaction, which is a chemical dialogue.
What is chemical dialogue?
They'll talk in chemicals. They're like they will exchange certain chemicals, or the release of certain chemicals will happen, because of which the pollen tube germination will take place. Okay. Now, but before that, the pistil, which is the female reproductive unit, it has the ability to recognize the right type, which is the compatible pollen, whether like there are the plants are present in the wild, right? They're not protected in the nature. So they have their own mechanism. So one such very, you can say, unique mechanism is that there is a possibility that many other kinds of pollen land on the wrong type of stigma. Now, why does it not lead to fertilization? Is because the pistil knows which is the right pollen, which is the wrong pollen. So it knows which is the compatible pollen. It can differentiate whether the pollen is of the right type, means compatible, or whether it is the wrong type, which is incompatible. After recognizing the right type of pollen, the pistil accepts the pollen. It's like, okay. Now, as a result of this, there is a continuous chemical dialogue between the pollen grain and the pistil. Chemical dialogue here is referring to the exchange of chemicals so that the pistil will know that the pollen is of what type. Okay. So, because of that particular reason, this is a part, like you can say, preceding the fertilization part. Okay. Now, after this, what will happen? The fertilization events. Now, see, the pollen tube enters into the ovule through the three regions. See, first, the pollen is going to land on the stigma, and then a chemical dialogue will take place, because of which the pollen tube will start growing and it will reach the ovule area. Okay, so a pollen is here, it will grow and reach the ovule part. Now, tell me one thing, students. In 60% of the angiosperms, the pollen grains are shed at what cell stage?
Two cell.
Two cell stage. What are the two cells that are present?
Generative and vegetative.
Very good. Now see what happens. Okay, which one is the bigger cell? Vegetative or generative?
Vegetative.
Very good.
Vegetative.
Very good. So now the generative cell. Okay, the pollen has two cells. The generative cell will start dividing. Okay, and it will form two male gametes. Okay, so initially the pollen was of two cell stage. Now, after the division, the pollen becomes three cell stage. Clear? Like this cell stage is what 40% of angiosperms pollens are released. But for the 60%, for the majority of them, the division takes place only when the pollen tube generation takes place, right? Now. Okay, now I told you that the pollen tube will reach the ovule part, the embryo sac. Okay, because ultimately the egg is there only. So it has to go there. Now, there are various places from which the pollen tube can enter. Now, if the pollen tube enters through the micropyle, that pore area where integument was absent, micropyle. So when that is happening, exactly, that is happening, it is known as porogamy. Okay, see, I told you there are so many gametes here. And if the pollen tube is entering through the chalaza side, then that is known as chalazogamy. If the pollen tube is entering through the integuments area, integuments, you remember, protective covering surrounding the nucellus, that is known as mesogamy. So it can enter through any of the area. Clear? But remember one thing, that even if it enters through micropyle or chalaza or integument, it has to come to the synergids only, no matter where it is coming from, because the egg is there, right? Now, the filiform apparatus of the synergids guides the entry of pollen tube, because we know that is their function. Now, after entering one of the synergids, the pollen tube releases the two male gametes in the cytoplasm of the synergids. So, filiform is helping the pollen tube to get inside. Once it had gotten inside, it releases the male gametes in the synergids. Now, one of the male gametes moves towards the egg cell and fuses with it. This results in the formation of a diploid cell, which is known as zygote. One male gamete, right? One male gamete plus one egg is equal to zygote, which is diploid. Clear? This first part is clear? Now, this process wherein one of the male gametes fuses with the egg cell is known as syngamy. Syn means fusion. Gamy is referring to the gametes. So the fusion of gametes is syngamy. Now, the other male gamete moves towards the two polar nuclei located in the central cell and fuses with them to produce a triploid primary endosperm nucleus. The second male gamete, it will go and fuse with the two polar nuclei. You people remember, right? In the middle of the embryo sac, this is the embryo sac. Here are the antipodal cells. This is the two synergid and this is the egg cell. And right in the middle were the polar nuclei, right? And this big part was the central cell. So one of the male gamete which came and fuses with this and forms a zygote. The other will come out here and fuse with these two. But before it fuses with these two, the two nuclei will fuse together and it becomes one. Okay? So N plus N, it becomes diploid. Clear? Now the male gamete is coming and fusing it with it, and then it becomes 3N, triploid, isn't it? 2N of the nuclei and 1N of the male gamete. So it fused and formed a 3N situation, and that triploid, just like how after the syngamy, a diploid zygote was made. After this fusion, a triploid, which is 3N primary endosperm nucleus is formed. And this process is known as triple fusion because three things are fusing. Okay, two polar nuclei and one male gamete. Is it clear? So, so these two,
Can you explain the triploid part again?
huh?
How it
Triploid?
became 3N.
See this. Even if, okay, first of all, you tell me if in the central cell there were two polar nuclei, what will be the ploidy of them? Ploidy of the
2N.
2N. You are understanding how it is becoming 2N because N and N, right?
Yeah.
Two nuclei are there, so N of both will become 2N. Now the male gamete, that is the other male gamete that is coming, it is also N. So 3N,
Oh,
this
okay.
triploid. And this cell, just like how by the fusion of male and female gamete, a zygote is formed, same way, when the male fuses with the polar nuclei, it forms an endosperm, primary endosperm nucleus. Because right now there is no cell, it was just nuclei only, right?
Yeah. Yeah.
So this process, the second type of fusion is triple fusion. The first fusion was syngamy. So in one embryo sac, two types of fusions are taking place, syngamy and triple fusion. So it is altogether known as double fertilization because two times the fusion is taking place. Clear? So when you sum total the syngamy along with the triple fusion, you get double fertilization. And this is the most unique feature about angiosperm. No other plant, algae, bryophyte, none of them have this particular feature, the presence of double fertilization. It is very unique and it is a very, you can say, advanced thing that happens only in the end. So double fertilization is clear to everyone? How it happens, what are the structure? Okay, so this is our fertilization event. Now we'll talk about the post-fertilization. But till that, note down up till here first. Or if you have any doubts, you can ask me, okay?
No doubt.
No doubts? Good. No doubts, then very good. You people can note down.
Done.
Everyone's done?
Yes, ma'am.
You can draw the diagrams, this rough diagrams, so that you don't get confused later on.
Done, ma'am.
Everyone's done?
Yes, ma'am.
Almost.
All right.
Done, ma'am.
Everyone's done?
Yes, ma'am.
Done, ma'am.
Everyone's done?
Yes.
All right.
Done, ma'am.
Everyone's done?
Yes, ma'am.
Done.
Everyone's done?
Yes, ma'am.
Done.
Everyone's done?
Yes, ma'am.
Done.
All right. So this was all about the fertilization event. Now moving forward, we'll talk about the post-fertilization events. Now we know that the meaning of post means after, the later one, right? So after the fertilization has been done, let us see what all things happen. Now see, in a nutshell, endosperm development takes place, embryo development takes place, ovules mature into seeds, and the ovary mature into fruits. Now we'll discuss each one of them. The first is endosperm development. Now, do you remember? As a result of triple fusion, primary endosperm nucleus was formed, which was 3N, right? Triploid. Now, those two nuclei that were situated so close to each other, they will fuse and form one. And that becomes the primary endosperm cell. So, the primary endosperm nucleus gets transformed into primary endosperm cell. Then, this primary endosperm cell, it divides repeatedly and forms the endosperm tissue. Again, endosperm tissue will also be 3N. So all of it is like dividing and getting, you can say, modified in one way or the other. Now, the cells of this tissue, meaning the cells of the endosperm tissue, are filled with reserve food material and are used for the nutrition of the developing embryo. The endosperm tissue that is formed later on, this is very important for the embryo to develop because it provides the nourishment. Okay. Now, the primary endosperm nucleus undergoes successive nuclear division, meaning karyokinesis, to give rise to free nuclei. See, normally we have learned in the chapter that is cell cycle and division that whenever a karyokinesis takes place, it has to be followed by cytokinesis, right? Then only it will result into the formation of separate cells. But if the cytokinesis is not taking place, then a multinucleated or syncytium, remember syncytium? Syncytium or syncytium situation will be formed. So talking about one kind of an endosperm wherein this happens, the primary endosperm nucleus divides and divides, means karyokinesis is taking place. But cytokinesis does not take place. So that type of endosperm which is formed is known as free nuclear endosperm. But when the the karyokinesis is followed by cytokinesis, and that results into the formation of a cellular endosperm. So, two types of endosperm formation can take place. It can be either free nuclear endosperm or it can be cellular endosperm. Now, afterwards, the cell wall formation takes place, and now the endosperm will become cellular endosperm. See, initially, when the cell wall was not formed, cytokinesis did not take place. So, that type was free nuclear endosperm, meaning one cell had multiple nuclei present in it, which none of them were surrounded by any wall. But when the wall started to surround them like this, wall formation took place and then it became the cellular endosperm. Clear? But there is one example, that is the coconut, right? The coconut water that we drink, it is the free nuclear endosperm of the plant. I mean, in that coconut, the coconut water that we are drinking, that is free nuclear endosperm. It is a nutritive tissue. That is why it is so high in nutrition that many a times it is advised to drink this in the summertime, right? But talking about the white kernel part, have you people had the white part of the coconut? It's very tasty, right? That part is the cellular endosperm because it is very thickly formed. Okay, one more thing. Have you people noticed that in those type of coconut that are brown on the outside, the ones that are broken, right? Do you see that how hard the white portion is that we consume? And it has very little water in it, isn't it? So that white kernel part is the cellular endosperm. The water of the coconut is the free nuclear endosperm. This is the example which contains both the type of endosperms in it. Clear? Any doubts up till now?
No, ma'am.
Okay. All right. Good. Now see, as I told you that endosperm has a function. What is the function? Endosperm has the function to actually provide nourishment to the developing embryo. Now, in seeds such as the pea, groundnut, and beans, the endosperm is completely consumed by the developing embryo. Okay, the endosperm gets completely consumed, meaning then after in these seeds, the endosperm is not left at all. In seeds such as the castor and coconut, the endosperm is present and it has not been consumed. So from seed to seed, it will vary whether the endosperm has been consumed completely or is it present. Up until now, everything is clear? Okay. This same portion about the seed will come when we'll talk about the seed development. But up until now, this is what you must know. So you people understand how the divisions are taking place for the formation of endosperm. What is the function of endosperm? Everything is clear? What is the function of endosperm?
It reserves food material.
It is the reserve food material and it is responsible for the nourishment of the developing embryo. Okay, that is the main function. Now, the second post-fertilization event that takes place is the embryo development. Embryo develops at the micropylar end of the embryo sac where the zygote is situated. Now see, the triploid PEN form the endosperm. The diploid zygote will form the embryo. Most of the zygotes divide only after some endosperm has been made, so that the developing embryo gets the nourishment. The early embryo development is known as embryogeny. Embryogeny is embryogenesis, right? The formation of embryo. Now, the process of embryogeny, it is same for both monocots as well as dicots because ultimately both of them are angiosperms. So, the formation of embryo is same for both of them. Now, there are certain stages which comes for the development of embryo. Now, see, if you look into your books, or have you people gotten your books here?
Not yet, ma'am.
Not yet? Oh.
No, ma'am.
Why are they not available, or the school is not has not notified you anything about?
Ma'am, over here it's not available yet.
Okay. Same for you, Mariam. It's not available. Okay, so only Yahya.
Now we have holidays.
Oh, okay, okay. Oh, now you people will get holiday for Eid, right?
Yeah.
Yes, ma'am. It is holidays right now.
Oh, you people are on your holidays.
Yeah.
Yes, ma'am.
Oh, good. Nice. Anyway, see, up until the formation of a mature embryo, there are certain stages that take place. The first is the pro-embryo stage. Pro means very initial part. Then that pro-embryo develops into globular embryo. A globular structure stage comes. Then comes a stage that is heart-shaped embryo. It sort of looks like this. Heart-shaped embryo. Then ultimately it leads to the formation of a mature embryo. So these are the stages that the embryo has to go through up until to become a mature embryo. Now talking about the embryo of a dicotyledonous plant, so it has two things. It has an embryonal axis and attached to that axis, it has two cotyledons because it is dicot. So this is an embryonal axis. Attached to it will be presence of two cotyledons because it is dicot. Okay. Now, the portion of embryonal axis above the level of cotyledon, above the level, meaning if cotyledon is here, so this portion above the level of cotyledon is known as epicotyle. Epi means up, right, on, and cotyle is referring to the cotyledon. And the region which is below the level is known as hypocotyle. You remember hypogynous, epigynous, perigynous? Remember? So epi means up, hypo means below. So these are the two regions. Okay. Now there is a reason why they are present. See, for example, this is an embryonal axis. Attached to it are the two cotyledons. Now the portion above the embryonal axis, like above the cotyledon area, this will be known as epicotyle. It is the name of the portion. Okay, the region is known as epicotyle. And the region below the level of cotyledon will be known as hypocotyle. We are not talking about any organ. We are talking about a region. Say.
In hypogynous, it was different, right?
Yeah, I mentioned hypogynous, epigynous because of hypoepi, not gynous part. I am referring to the position. Epi means on, like on top or above. Hypo means below. Clear? Now, the epicotyle, that region which is above the level of cotyledon, it will terminate with a plumule. It is again a region. Okay, plumule is a region which will give rise to the stem or the shoot, because we use the seed for agriculture, right? Whenever we want to grow a new plant, we sow the seed. Inside the seed, embryo is present, right? So that embryo has to have something, then only the roots will be formed, then only the shoots will be formed. So this plumule, it will give rise to the shoot or the stem. Same way, the hypocotyle, it will terminate with a radicle and it will give rise to the root. So in a seed, you will find plumule and a radicle, because of which when you sow that seed in the soil and you water it, then the radicle will give rise to the roots below the soil and the plumule will give rise to the shoot above the soil. Clear? Always remember that fruit, inside the fruit, seed is there. Inside the seed, embryo is there. Inside the embryo, endosperm is there. So when we are talking about all of these structures, it is from the inside to the outside, isn't it? Because fruit is the outermost covering, right? Okay. Also, many a times people get confused as to which part gives rise to what. Like plumule gives rise to shoot or root. So always remember, radicle starts with an R, root starts with an R. So radicle will give rise to root. A very short trick to remember this. Okay. Now, the tip of the root is covered with a root cap. Now, everyone remembers root cap from morphology. Its function was to protect the root, right? Now talking about the monocot embryo. Monocots. Mono means one. These type of plants, they have only one cotyledon. Just like how dicots had two cotyledons. Cotyledons, have you people seen the peas in a pod pea? Right? Everyone
Yes.
has the garden peas. Now, if you remove the outer layer of that, two halves you will get, isn't it? Two halves. Those two halves are cotyledons. Okay, those two halves are cotyledons. So just so you know, now if you try to split open the seed of a rice, has anyone seen the seed of a rice? No.
No, ma'am.
Okay. Just to let you know. Okay, wait. Just 1 second. Give me 1 second. Okay. See this. Has everyone seen this?
Yeah.
This is the seed of the rice seed. Okay. Now, by just looking at it, can you think you can split it into two parts? No, right? It seems impossible. It is not even possible at all. Because of this very reason, it is a monocot. Monocot means only one cotyledon is there. You cannot split it. Okay, now if we search the seed of P, everyone has actually tried to split this, right? Once in your lifetime, I think everyone has. When you,
Yes,
okay, leave this. Have you seen kidney beans, rajma, right? Everyone has had that. Okay, wait, that is a better example than this. Kidney beans, that is rajma. Now, if you take a rajma and you try to split it, you can do it, right? Everyone has, like, you might have felt it during eating it also, that it got split into your mouth. So that is a dicot. Whenever a seed can be split into two parts without any, you can say, using anything, scissors or anything, that is a dicot. When it cannot be, then that is a monocot. This much is clear to everyone? All right. Now talking about the monocot. See, now you understand what I'm saying by dicot and monocot? Okay. Now talking about the monocot embryo. So the number of cotyledons will be 1. The name of the cotyledon in the grass family is known as scutellum. They have kept the name of the cotyledon as scutellum. Now, if you look at the seed of this thing, rajma beans, the kidney beans or the peas, you will find that if you hold it like this, the two seeds will be present in the middle, right? But for this thing, the monocot, the cotyledon, whichever is there, it's lateral, sideways in position. It's not in the middle. Okay. So the name of the cotyledon, that one cotyledon, it is so special that it has a name. The name is scutellum. Is situated towards one side, meaning lateral in position of the embryonal axis. See, the two cotyledons in the case of dicot, it was situated on the either side of the embryonal axis, right? But in this case, it will be on one side. Clear? Sort of like this pointy part. Okay. All right. Moving forward, radicle is present, just like how dicots also had radicle, and root cap is also present. Now see, the radicle and plumule, both of the structures are present in the monocot seed, but the radicle has a protective layer on top of it. Okay, a layer sheath which is known as polyorhiza. Polyorhiza. It will enclose the radicle, meaning radicle is present, but it is enclosed by a sheath. And that sheath that encloses the radicle is polyorhiza. It is sort of like a blanket. Okay. Now talking about the plumule or the shoot apex. See, the other name for radicle is the root apex. The other name for plumule is the shoot apex because it gives rise to root and shoot. Okay, so plumule is also present, and the layer which covers the plumule is known as coleoptile. Again, to avoid the confusion, coleoptile has P in it, so it will cover plumule. Polyorhiza has R in it, so it will cover the radicle. So the endosperm development and embryo development, both of these topics are clear to you people? You understand what is this whole shebang about? The seed,
yes,
the cotyledon and everything? Any doubts, anyone? All right. See, we are almost nearing the end of the chapter. Almost. It's like half of another class and we're done. Okay. And then we'll start a better chapter that is human reproduction that everyone can relate to, right? Because it will be human related structures, right? Human related stuff. Anyway, okay, first you note down up till here. Are you sure you people have no doubts? This whole thing is clear about the seeds?
yes, ma'am.
All right, very good. You people are very good. You don't make me work very hard. You can switch off your videos and write comfortably. It's fine.
Done, ma'am.
Everyone's done?
Yes.
Done, ma'am.
Done, ma'am. Done, ma'am.
Everyone's done?
Yes, ma'am.
Done, everyone?
Yes,